Hey, it's Group 5. Let's talk about lipids.
He knows a lot about the science stuff, Professor Dave explains.
Lipids are a diverse group of organic compounds including fats, oil, and waxes, characterized by their insolubility in water. They serve as concentrated energy, providing more than twice the energy per gram compared to carbohydrates, and are essential components of cell membrane, forming the lipid bilayer that regulates cellular transport. Steroid, a type of lipid, act as a hormone regulating various physiological process. Furthermore, lipids provide insulation and protection that are crucial for absorption of fat-soluble vitamins.
As for our methodology, we have the materials and reagents that will be used for the experiment. For the apparatus section, we need aspirator, aluminum foil, beakers, Bunsen burner, evaporating dish, graduated cylinder, hot water bath, iron ring and stand, matches or lighter, pasture pipette, serological pipette, test tube holder, test tube rock, test tubes, and wire gauze. For the reagents, we need our solvents, which are the 2 moles of sodium hydroxide, butanol, ethanol, hexane, distilled water, sodium carbonate, and dishwashing liquid. And for the samples, we need canola oil, olive oil, unknown 1 and unknown 2, and palmitic acid. Other reagents are also needed, such as the H2SO4 or sulfuric acid and iodine solution that will be used for the Hubble's test in identifying unsaturated lipid samples.
So the preparation of samples and solvents is we need 5 ml of each sample, unknown 1, unknown 2, canola oil, and olive oil, and approximately 1 gram of palmitic acid and place it in a vial. For the solvent preparation, we need to measure 5 ml of each solvent into separate vials, water, NaOH, butanol, ethanol, and hexane. So the first test that we are going to do is a solubility test. We are going to determine the solubility of different lipid samples in various solvents. First, we need to label 5 test tubes and dispense 1 ml of each solvent, water, NaOH, butanol, ethanol, hexane into each test tube, and add 10 drops of canola oil sample to each test tube. Shake each test tube well for 1 minute and allow the mixtures to stand for 2 to 3 minutes, and observe if the mixture separates into distinct layers. Repeat the process for the other samples, olive oil unknown 1, unknown 2, and palmitic acid. For palmitic acid, use only a small pinch. So the next test that we are going to do is the Hubble's test. It helps us to identify the unsaturation of lipid samples using iodine solution. So we need to label 5 test tubes and add 10 drops of each sample, canola oil, olive oil, unknown 1, and unknown 2. And add a pinch of palmitic acid in the last test tube. Add 1 ml of hexane to each test tube and mix the contents of each test tube well. Add iodine solution dropwise to each test tube, stirring after each drop until a pink color appears. Record the number of drops required for the appearance of the pink color in each test tube. Count the number of iodine drops needed to reach pink color. More drops typically indicate higher levels of unsaturation. So the third test that we are going to do is a saponification test. It assesses the presence of ester bonds in the lipid sample by testing for soap formation. So first, we need to label 5 test tubes and place 0.5 ml of each sample, canola oil, olive oil, unknown 1, and unknown 2 into separate test tubes, and place a pinch of palmitic acid in the last test tube. Add 1 ml of 2-MeNaOH and 0.5 ml of ethanol to each test tube. Cover each test tube with aluminum foil and heat the test tubes in a hot water bath for 15 minutes. After heating, allow the test tube to cool slightly and add 3 ml of distilled water to each test tube. Shake each tube for 30 seconds and observe for the formation of froth. And if soap is formed, frothing will be observed due to saponification. So the next test is the emulsification test. It tests the ability of lipid samples to form an emulsion with water and different emulsifiers. So first, we need to label 5 test tubes and add 10 drops of each sample: canola oil, olive oil, unknown 1 and unknown 2 to separate test tubes, and add a pinch of palmitic acid in the last test tube. Add 1 ml of distilled water and 5 drops of sodium carbonate solution to each test tube. Shake each test tube for 2 minutes and observe immediately for the formation of an emulsion. For the last procedure, we are going to repeat the process, but we are going to replace the sodium carbonate with dishwashing liquid as an emulsifier. And an emulsion is formed if the lipid disperses evenly in the water, creating a cloudy solution.
Moving on to the results and discussion, under this is the pre-lab and post-lab questions. For the pre-lab question 1, what characteristics of lipid differentiate them from other biomolecules? Unlike other biomolecules, lipids do not have a common structural feature that serves as a basis for defining such compounds. Additionally, lipids are hydrophobic, which is one of their primary characteristics. They are largely insoluble as a result of their poor water solubility, while proteins and carbohydrates have a high tendency to dissolve in water. For the second question, what are saturated and unsaturated fatty acids? How does saturation affect their physical properties? These fatty acids are simple lipids containing a long chain of carboxylic acids and alcohols. Specifically, saturated fatty acids are those where all carbon bonds are single bonds, making them saturated with hydrogen atoms. Due to the lack of double bonds, there is more packing of chains, therefore more intermolecular forces to break, leading to a high melting point. On the other hand, unsaturated fatty acids are those that contain one or more double bonds in their hydrocarbon chain. Due to the presence of double bonds, there is less packing of chains, thus less intermolecular forces to break, leading to a low melting point. This explains why saturated fatty acids are solid at room temperature, like butter, while unsaturated fatty acids are liquid at room temperature, like vegetable oil. For the last prelude question, what is emulsification? What are the two types of emulsions that can be formed? Emulsification is a process of combining two immiscible liquids into a stable one. The two types of emulsion are oil in water emulsion or direct, and water in oil emulsion or reverse. Direct emulsification is where oil droplets are dispersed in continuous water phase, while reverse emulsification is the other way around, where water droplets are dispersed in continuous oil phase. After performing the qualitative tests for lipids, the physical changes were observed, and the results were tabulated. For palmitic acid, it was observed to be soluble in nonpolar and insoluble in polar solvent, which is in line with the expected result since it is a lipid. In Newell's test, it took 26 drops before the color turned pink, making it the third most saturated sample among the 5. Meanwhile, it tested positive for saponification test, indicating that it reacted with NaOH to form soap and glycerol, which is in line again with the expected results. It also tested positive for the emulsification test under sodium carbonate, while negative under the dishwashing liquid. For olive oil, it was observed to be soluble in nonpolar and insoluble in polar solvents, which is again in line with the expected results since it is a lipid. In Newell's test, it took 23 drops before the color turned pink, which is in line with the expected results since it is an unsaturated fatty acid. Meanwhile, it tested positive for saponification test, indicating that it reacted with NaOH to form soap and glycerol, which is again in line with the expected results. Lastly, it tested positive for the emulsification test both under sodium carbonate and dishwashing liquid, indicating that these are effective emulsifying agents for olive oil. Moving on to the next sample, which is the canola oil, it was observed to be soluble in nonpolar and insoluble in polar solvents, which is in line with the expected result since it is a lipid. Meanwhile, in Ubel's test, it took 20 drops before the color turned pink since it is an unsaturated fatty acid. Meanwhile, it tested positive for saponification test, indicating that it reacted with NaOH to form soap and glycerol. And lastly, it tested positive for emulsification test both under sodium carbonate and dishwashing liquid, which also tells us that it was an effective emulsifying agent for canola oil. Moving on to the next sample, which is the unknown one, at solubility test, the sample was insoluble in organic solvents, suggesting that it is polar. For Ubel's test, it indicates that it is an unsaturated fatty acids since it took 43 drops before the color turned pink. Meanwhile, for saponification test, it tested negative, ruling out the presence of esters. And for emulsification test, it was negative under sodium carbonate but positive under dishwashing liquid, indicating that it has some surfactant properties. Given these observations, the sample could be an unsaturated fatty acid or a related compound that is water-soluble, such as sodium or potassium salt of a fatty acid. Meanwhile, the observations for the unknown 2 is that it was soluble in nonpolar. It took 28 drops for the color to turn pink. For the saponification test, it tested positive. And lastly, for the emulsification test, it tested both positive under sodium carbonate and dishwashing liquid. Given the results of the tests, the sample is most likely a fatty acid ester, specifically an unsaturated triglyceride. Moving on to the post-lab questions. Question number 1. Below are 3 lipid samples. Which tests in the experiment can be used to differentiate the 3? The tests that can be used to differentiate these 3 lipids are the Ubel's test and the saponification test. In terms of the Ubel's tests, sample C and sample B will test negative due to the absence of double bonds, while sample A will test positive due to the presence of double bonds. In terms of the saponification tests, sample C and sample A will test negative due to the absence of esters, while sample B will test positive due to the presence of esters. Therefore, it can differentiate triacyl glycerols from both saturated and unsaturated fatty acids. Moving on to the question 2. Margarine is solid at room temperature due to hydrogenation, a process that adds hydrogen atoms to unsaturated fatty acids in plant oils, breaking some double bonds. Then this increases the proportion of saturated fatty acids, which are more tightly packed, making the margarine more solid. For the last post-lab question, to answer, trans fats increase your LDL or the bad cholesterol, which is why they are bad, while they lower the good cholesterol or the HDL. A buildup of cholesterol in our arteries or the blood vessels can be caused by low HDL and high LDL levels. Your risk of stroke and heart diseases rises as a result. When it comes to heart disease and stroke, polyunsaturated fatty acids can help lower blood levels of harmful cholesterol. They also supply nutrients that support the growth and function of our body cells.
And as for conclusion, a valid technique for determining whether lipids are present in a sample is to use qualitative testing. We can accurately determine the lipid content of different substances by employing methods like solubility tests, transparent spot tests, acrolein tests, and more. Although these tests are useful tools, It is essential to remember that for thorough examination of lipid composition, they should be used in combination with other analytical methods. Researchers can better understand the function of lipids in biological systems and their applications in a variety of sectors by combining qualitative and quantitative methodologies. And for our recommendations, the group suggests improving the following: making sure that there are specific pipettes, beakers, and/or dispensers for reagents and test samples to prevent cross-contamination. If required, dissolve the lipid in a suitable solvent such as ether or chloroform before testing. And since many chemicals, particularly organic compounds, are prone to degradation over time, and for this, it is best to use fresh reagents to prevent degradation and loss of reactivity. This may result of processes like photodegradation, hydrolysis, or oxidation. Lastly, chemical reactions often require specific ratios of reactants to proceed efficiently. Using the correct number of reagents ensures that these optimal conditions are met.